what was that? Right after exam 2, we pretty much just went through and looked at facts. Okay? We did some redressing of how you go through and you name the whole alpha, beta, 1, 4, all that fun jazz. I thought about putting a question on that and decided against it. Okay? Just figured we'll stick with the facts off of this. So, cellulose. Lots of fun little facts on here. What might you take away? What kind of question could I ask about cellulose? Let's try it that way. Any ideas?
The beta 1,4 glycosidic bond.
Okay, we could be referencing that beta 1,4 glycosidic bond, which is interesting for what reason?
They just don't process them.
We don't, as humans, don't have the, in fact, as animals, don't have the ability to break the beta 1,4 glycosidic bond. Okay? So what does that mean? We can't digest it. So cellulose is a nice fiber process that just comes into our digestive system and goes right back out.
It's like just fiber.
Yeah, it's effectively,
But that's why it's
kind
good
of.
for structure.
What's that?
It's good for structure because it doesn't break it.
It doesn't break, yeah. So that's going to be the other aspect. When you look at cellulose, it's great for structure because we don't break it down. We can't break that down. And that would kind of suck if we could break it down because then it doesn't work very well for structure.
Doesn't it help us produce new bile because of the fibers?
That's a good question. I don't know on that.
With the liver?
So I would say we should not worry about that right now because that's definitely not on the test. But yeah, the important aspect is that we're looking at that beta 1,4, which is not processable. Oh, come on. Something's got to work today. Starch. So one of our storage mechanisms is starch, so we can look at amylose and amylopectin. Those are kind of neat because we can actually identify those compounds with iodine. So iodine will coordinate to the sugar molecules in that nice little helix, and you get some cool color changes coming out of that. Okay. I'm less concerned about this slide than I am about this slide, which is how we break those down. Okay, so it's helpful to know what we're talking about with those. So your amylases are the enzymes that go through to break your starch molecules. So if we look at our different starch molecules, we've got all those fun little linkages in there, and we want to be able to come up with some way to go through and break those. Okay. And since we have different types of connections within your starch, you need different types of enzymes to go through and break it down. And so there were two enzymes that we referenced, the beta amylase and the alpha amylase. And the more important aspect with that is that their characteristics. So the beta amylase was an exoglycosidase. So that ACE ending sort of means, it's a signifier that we're looking at an enzyme, and it sort of means looking at breaking something apart. So when we're looking at an exoglycosidase, what we're doing is breaking glucose bonds away from each other, or glucose structures away from each other that were connected through glycosidic bonds. Okay. So if we looked at our primary difference between these, your alpha amylase is an endoglycosidase as opposed to an exo. What does that cause as a change between these two enzymes? What might we see as a difference?
Where it breaks.
What's that?
Where it breaks.
So where it breaks is going to be an important aspect. We aren't going to be able to visualize that as much. So how would we end up visualizing that? We see a rate difference.
Mm-hmm.
Okay. So your beta amylase being an exoglycosidase can really only break things apart on the outside of the structure. So what you're looking at for a concentration, so if we think back to our rate laws as a general concept, your rate is equal to your rate constant times the concentration of one of your reactants. Okay, close enough. If we're looking at an exoglycosidase, what you're talking about is only reacting on the outside. So your concentration is now dependent on what's on the outside of your starch. If we then shift to the endoglycosidase, that can now cleave anywhere within your starch molecule, which means the effective concentration has done what? Increases, which means what happens to our effective rate? also increases. Okay, so that's one of our big things that we can notice between those two. Chitin and ultimately your bacterial cell walls. Right? What is something that both of them have in common? Okay, I'm going to try and not do this obnoxiously. So there's chitin. Give you some time to take a look at that image. And your cell walls. What do they have in common?
Same glycolytic bonds.
We are looking at similar glycosidic bonds, but then wouldn't we expect the same result as we would get for starch? And starch is supposed to be an energy storer, and our bacterial cell walls and chitin are not just energy storage. They contribute to structure. So what else has changed about our glucose molecules in these cases to contribute to structure? Yeah, we've got that amino group. Oh, don't move. Dang it. We've got that amino group down there. How is that going to contribute to structure? Yeah. What we do is greatly increase the amount of hydrogen bonding between those. Oh, that was a bad idea. I'm going to sit back down. But I want to move, so I'm going to wheel around. What we've done is greatly increased the amount of hydrogen bonding between those chains of our glucose molecules. That increased hydrogen bonding means our chains are now stronger linked to each other, which then means we get a more rigid overall structure, and we can contribute to structure or shape a little bit better. Okay? So both chitin and your bacterial cell walls are following that similar kind of pattern. We're just increasing that hydrogen bonding interaction. It's kind of a fun little fact about chitin, just because I was interested in it and started digging around a little bit. It contributes to structure, just like cellulose, but we can decompose chitin. Okay? We actually have chitinases, okay? ACE for the enzyme and chitin now, so it's an enzyme that will actually degrade chitin. Okay? So we can actually get energy out of the exoskeleton of our insects. Last thing that was kind of interesting out of that chapter was looking at our glycoproteins. Oh, I can't get around. Within our glycoproteins, we were talking about the biggest example being our A, B, and O, our blood types. Okay? And all of that is due to a carbohydrate chain hanging off the back end of our cells. Okay? So our body then recognizes that that carbohydrate chain and doesn't attack it. So if we get a foreign blood type into our sample or foreign plasma, we can potentially have a different carbohydrate chain. Your body will or the immune system will recognize that as a foreign body and will attack it. Okay? So that's kind of a neat process out of all of our carbohydrates. Our A, B, and our blood type system is actually based off of carbohydrates, and we're just looking at small polysaccharides hanging off the end of our cells. It's kind of a neat thing. Now we're officially moving into chapter 15, where we look at metabolism. This breaks down into two processes, catabolism and anabolism. There is a question on sapling that was difficult to understand because of how it was phrased. It is a very important question. I would recommend you go back and evaluate that one, because what it's looking at doing is, in a catabolic process, what things do we expect to happen? In an anabolic process, what things do we expect to happen? So what's coupled with each of these things, or in those processes? Okay, so in catabolism, what we're doing is breaking large molecules. Okay, so a nice example for that is we could break glucose down into CO2, ultimately. What was the process, or what did that do when we catabolized glucose into CO2? We released a bunch of energy. Okay, where did that energy go? Into the formation of ATP. Okay, so it's looking at what are each of those processes doing and where's the energy coming in and out of. Okay? In catabolism, you're transferring electrons to acceptor molecules, and your overall process is one of oxidation. In anabolism, you're pretty much looking at the reverse. We take small molecules and we build them up, which means to build them up, we have to provide the energy to do that. That energy is going to be supplied from ATP. So instead of making ATP, we'll consume ATP for that process. And we're looking at acceptance of electrons from transfer molecules, so we're looking at a reduction process, okay, for anabolism. And the reverse of that, plants can take CO2 and turn it into glucose. Okay? So it's looking at those individual processes. Should have removed all the steps out of these. Since we are looking at the transfer of electrons, particularly in this system, we want to have some understanding of oxidation and reduction. However you remember that is going to be works for you. Okay? I tend to go with the Leo Says-Gur, loss of electrons, oxidation, gain of electrons, reduction. That is the strict way to evaluate oxidation and reduction. There were shortcuts. What were some of those shortcuts? So we can look at oxidation being one of increasing oxygen bonds. Okay. The stricter definition that you would get from organic chemistry is not just the increase of oxygen bonds, but increasing the amount of, what was that?
Protons?
No, not quite. Electronegative atoms. Using electrons we could use, but that comes out of looking at the oxidation state. So you could go through and calculate all those. That's a lot of work. So what we want is something kind of similar. So if we or easier to work with. So if you increase the oxygen bonds or increase the amount of electronegative atoms, okay, you're looking at an oxidative process. Conversely, you could look at the number of protons, okay, and we're focusing around a particular carbon atom. We could either increase the number of oxygen or we can decrease the number of hydrogen bonds. Okay, that ends up being one of our easier comparisons, is just looking at what changes. Do we increase the number of oxygen atoms? Oxidation. Okay. Did we decrease the number of hydrogen atoms? Oxidation. Okay. So depending on what type of reaction you're doing, you may decide to use different definitions. Because there are cases where you may change the number of hydrogen atoms and it does not appear that you've changed oxygen. Okay? You will have changed the number of electronegative atoms, okay, or the bonds to electronegative atoms. Okay? So depending on what you want to look at, that can help you decide what's going on with oxidation. What if we switch this to reduction?
Decrease in the number of electronegative atom bonds and increase in hydrogen bonds.
So if we switch this to reduction, we now need to be doing the opposite. Well, if oxidation was increasing oxygen bonds, our reduction is going to be decreasing. And if oxidation was decreasing hydrogen bonds, reduction will be increasing our hydrogen bonds. Okay. When we say hydrogen bonds, we're not referring to hydrogen bonding. This is an actual bond change, okay, a physical, chemical, or sorry, a chemical change. We've got our primary molecules that go through and deal with this exchange. NADH is the probably more important one of our electron carriers because of its relatively small size. It is fairly mobile, and it can transfer electrons from one part of your cell to another part of a cell fairly easily, at least in comparison to other transfer mechanisms. What we talked about in class was what's happening within that. So we are coupling usually some kind of oxidation or reduction pathway with our NAD plus NADH reaction. Okay, so if we take a look at our system going from ethanol to acetaldehyde, what type of process is that? Okay, so what we would do is focus on what happened to our carbon. Okay, it gains a second bond to oxygen, and it loses a bond to hydrogen. okay, which means we did oxidation. Okay. Remember, we refer to them as redox because we never have oxidation on its own, we never have reduction on its own. Okay, so what has to then happen? If we're going to do that oxidation reaction, our NAD needs to go through and run the reverse. So if that's oxidizing, what does NAD need to do? Reduce. So to go through and reduce, what does it need to do? Accept electrons. Right? It's going to accept those electrons in the form of hydride, which is ultimately coming out of there. So that hydride now shares electrons with the NAD. So now the question is, which form does it share them with? Okay, so there's one option we can cheat. This is NAD plus. How do I know it's the plus? It actually says plus right there.
Oh,
The other way we could look at, and then NADH. How do we know it's not NAD plus? Well, there's not a plus right there. We have that electron pair. Okay. You are responsible for being able to look at those structures and say what the different states are. Okay. So what changed as we went from NAD plus to NADH? We accepted hydride. So we're actually looking at increasing the number of hydrogen bonds and ultimately decreasing the number of electronegative bonds. So we've increased the number of hydrogen bonds at that carbon, and we decreased the number of electronegative bonds. At that position. Both of those in turn lead us to reduction as our process. Okay. What ends up happening? Well, we can do our arrow pushing. Remember, it's hydride coming in as an H minus. Those electrons can come in and attack that carbon. How many bonds does that carbon now have? Too many, so we have to break a bond. What bond do we break? The polar pi bond. Now how many bonds does the next carbon have? Too many, so what happens? We break that pi bond and dump those electrons onto the nitrogen, which is why we have that lone pair now on that nitrogen. Okay? So having some understanding of how those electrons move is of critical importance, and looking at those structures and identifying. How do that make sense? Questions? Are you all saying this is too easy? Okay, just checking. I just didn't hear any questions. We did look at FAD, which is our other electron carrier. You'll notice it is significantly larger. And that size is one of the things that contributes to it being stuck within the enzyme. Right? So FAD is not very mobile. To get it as an electron carrier, you're taking it along with the entire enzyme that's holding on to it to wherever you want to dump those electrons, okay? Or technically steal the electrons from. So within this, we got to be able to recognize our pieces. Okay, so this slide goes through and shows that. We have AMP down below. AMP stands for adenosine monophosphate. So there's our single phosphate. Adenosine is in reference to adenine being our nucleotide bound to a sugar molecule. So that says sugar, and then this is, I may be saying it wrong. It may not be a nucleotide. We'll talk about that after the exam. Well, it's not super relevant to your question. Because we can call them nucleotide, nucleosides, and all sorts of other fun stuff. And I never remember those. Okay. But that's what makes this AMP. You've got the phosphate, and then you have the adenine aspect of that, which is a nucleotide. If we continue through the rest of our structure, that is then linked to ribitol. Right? If we think way back through our carbohydrate chapter, what we have here is a derivative of a carbohydrate, a sugar. Okay? The only difference is that you'll notice there is no carbonyl. Okay? So we're not looking at ketose or an aldose, we're looking at the sugar derivative, the alcohol derivative of it, and that's why we get the ribitol. That is then connected to, ultimately, the most important part of this, which is our isoallo, that word, which is where our oxidation and reduction is actually occurring. Okay. That is occurring where? Those two nitrogens are our key derivers of this chemistry. Depending on how you want to go through and address this, either as a radical mechanism or as a heterolytic ionic mechanism, you may go through and split that differently, but that's ultimately where your chemistry is happening, is up in that isoalloxyzine structure. Okay. And we're putting hydrogen there and a hydrogen to that nitrogen. If we go radical, we're looking at single electrons. If we go ionic, we're going a negative there, which means the other hydrogen comes in as a positive. Okay. Not hydride, proton. Hydride is top, proton is bottom. Okay. So it's just looking at those individual pieces and kind of referencing them. FMN as a term shows up fairly frequently. Okay. FMN is in reference to a mononucleotide. So the flavin is looking at your kind of piece up here, and within the flavin there is a version, a derivative of a nucleotide connected to a sugar, which is what happened with our AMP. We've got our nucleotide, or polynucleoside, connected to a sugar. Okay? So when we look at FAD, and it's a dinucleotide, that's what they're referencing in the dinucleotide. We have two, for lack of a better description, we'll call them nucleosides. Okay? Your adenine, guanine, cytosine, all that fun stuff that you vaguely remember from biology, connected to a sugar. That's your nucleotide. There are two of them in this structure, which is why we end up calling it FAD for D for the dinucleotide, as opposed to FMN for mononucleotide, because it's not including the AMP part of it. Kind of, sort of? Oh, I thought I deleted this. So we do have kind of the neater system where we're just looking at the reductive part of our molecule, where we're looking at bringing those electrons in. Okay? And this is how we could get really specific with it if we really wanted to do all the electron pushing, which I would say you shouldn't really stress that much about. Okay? We can have
So you're not gonna
that come out
the
to react
actual
with H+.
mechanism on the exam?
No, and we could have H- come in and attack here. And you end up shifting across to that final product. Okay? Or you could do it in a radical type fashion.
Would you really necessarily see the radical reaction in the biological system?
So there are radical reactions in biological
Sure.
systems. So the question is exactly how does the FAD do its process? The only way we would really be able to detect which way was the better mechanism would be isolating it out of the system. As soon as you isolate it out of the system,
Then it falls apart. It's not the
you
same.
are very likely not looking at the same process. You could look at it.
I know, I'm looking at the damn bars.
The other way you could do it is look at the whole system or most of the system and then throw in secondary species that would detect for radicals.
Sure.
So if a radical was formed, that could potentially react with our signifier, and we could see,
Could see it.
the radical form potentially. If you've got a radical detector, any radical that forms, it doesn't necessarily mean that it's coming from this. Okay. So that becomes a little bit trickier.
Mike,
Yeah,
I
sorry.
have a question. So what's the difference between the NADH and FADH2?
So our difference, the primary difference is our size, which translates to mobility. NADH, very mobile. FADH2, not mobile. Okay, that's our biggest difference.
Isn't the other big difference that NAD only accepts hydrogen, but the FAD can accept hydride and hydrogen?
So careful, NAD still accepts hydride.
Yes.
Both of them accept hydride. What ends
But
up
you
happening
end
in
with the hydrogen ion with
Yeah.
the NAD mechanism.
in the NAD coupling, let's see.
Can FAD accept them at the same time, though?
Yeah, it can. So in NAD, what happens is you only pick up hydride. You only pick up H minus. If we kind of balance these reactions out, what we would end up with, let's see if I can do this appropriately, is we'd have our alcohol plus our NAD plus. Okay, the hydride cancels, and I hate how they show that, but that's ultimately what this is. The hydride cancels out across the reaction, and we would get our carbonyl NADH. Okay, whoops, and H plus over here. So to do the conversion, you still have to get rid of that H plus. That H plus is ultimately coming from our alcohol. NAD can't accept that H plus. FAD, on the other hand, has two sites, depending on how we want to look at the chemistry. We can take the hydride as H minus, and we can share those electrons with the nitrogen. There's now too many electrons around the nitrogen, so we can take those two electrons and shuttle them into a second bond between our carbons. But now that bottom carbon has too many electrons, so we can shuttle those electrons onto the nitrogen. But the nitrogen now carries a negative charge. That's not stable, so instead of shuttling them directly to the nitrogen, we shuttle those electrons out to H plus. So FAD will take both the hydride and H plus. NAD can't take the H plus.
H plus stays in solution, that's why they show it on.
Probably picked up by the enzyme. And that's why they're showing it, because to do that reaction, so this is something that does bother me.
Balanced when you're.
Yeah, it's not, yeah.
But isn't that why it's so important in the citric cycle? Because if NAD and AD are so close, you need FAD in the cycle, but you need FAD because of its ability to pick up the hydride and the hydrogen.
Both of them contribute protons into the electron transport chain in one form or another. What ends up happening with NAD in its conversion over to NADH, and let's see if I can do this right. They almost always show this reaction as this,
Yes.
okay, with that H plus. So in the conversion of NAD plus to NADH, we are still forming H plus. Okay? So it is coming into the solution. That now changes the pH of the solution. The electron transport chain can grab H plus that's in the solution and shuttle it across. FAD directly transfers that into the electron transport chain, as opposed to here where it's now in solution and the solution is now taken care of.
Which then changes the concentration.
Changes the
proton
concentration, which is,
and then
yeah.
drives the.
Yeah. So that H plus is not just free floating. Okay? More than likely that was picked up by the enzyme, and then the enzyme spits it off onto water, and then it goes out into the solution. Whereas the FAD transfers it both with the hydride and the H plus. Those are on one structure. And so you could potentially look at maybe that being a more efficient one, because you're now not waiting for diffusion of the H plus. It's already bound. So you can, here you go. Here's both.
And it's just.
And I don't think you end up seeing an efficiency difference there. I'm not positive on that. But we could see if we've got totals somewhere further on in the slideshow.
I think NAD is producing more ATP than FADH2.
It does.
NAD produced 2.5 and I think FAD2 produced 1.5 or so.
1.5. So what ends up happening is the NAD actually does a better or more efficient transfer of it. Okay? Which you might say is counterintuitive, because it's dropping it into the solution, we're waiting for that diffusion. Thanks, biochem. Thanks, biology, officially. Yeah.
So, in general, you're saying FAD and FADH2 is not mobile, correct?
True.
Okay, so when you're producing FADH2, it's immediately transferring those hydrogen ions into the ETC?
Immediate, assuming the contact is there, yes.
Okay, so while you're going through
So
these-
if, so,
It depends on rotation, it depends on how it lines up.
let's see,
right?
do we want to skip ahead on this or not? Let's see if I can jump ahead. It's significantly further ahead. So the only place where we see FAD pop up, whoops, come back here, is in that succinate dehydrogenase, going from succinate to fumarate. That enzyme was located where?
Between those two?
No, in the cytosol.
True, in between those two, it's not in the cytosol.
Citric acid cycle?
It's in the membrane. Okay? This enzyme is part of complex 2 when we're looking at the electron transport chain. So it picks up that FADH2, and the FADH2 is now not very mobile, but we have to get those electrons into our electron transport chain. So what does it end up doing? It transfers those electrons to our Q cycle, which is now within the membrane. NADH doesn't have those electrons free floating in the solution, or sorry, NADH is not integrally linked. It's now free floating through the solution, and it has to bind to enzyme 1. Why do we have to bind to enzyme 1? Where's CoQ? CoQ's in the membrane. NADH doesn't make it into the membrane, right, as part of its kind of transfer. So the FADH2 does facilitate and help that reaction, and really we should be showing that.
I was gonna say, it's missing.
So,
It's
and
showing
so
all the NADH.
this is going to come from a perspective. Which do we care more about? If FAD doesn't move, it never leaves the enzyme, in which case does it make sense to really show the FAD couple here? Not really, because what ends up happening, ooh, let's see if I can draw the circle right. I don't know I can pull this off, but let's see what I can do. Okay. We have Q, we have, oh man, was it QH2? Crap. That whole brain damage thing. QH2. What ends up happening is to get that Q to go to QH2, what are you bringing in? Ah, man, come on.
Yeah, NADH2 is bringing up the hydrogen.
Oh, what's going to be the better way to couple this?
It's kind of off.
So to do this process, we have FAD come in to form FADH2. Man, they can do a much better job of drawing this than I can. Ah, here we go. FADH2 will reform FAD in the enzyme, but as that happens, what happens? Q goes to QH2. QH2 then goes on further on down the cycle. Okay? So the enzyme here really just transfers FAD to FADH2. FADH2 then reacts with our coenzyme Q to form our reduced form of coenzyme Q to shuttle the electrons further into the electron transport chain. Does that kind of make sense? So our FAD is locked, NAD is not. Okay, and now we got a long, dizzying flip back through. More questions on FAD? The last one that is talked about in this chapter is looking at ATP. ATP is just our transfer of energy. Okay. It's a fun one because we've got the phosphate there. So why do we end up seeing this being such a large release of energy? Okay. So we got ATP going to ADP plus PI. Why is energy released? It's not always necessarily the case. What's that? So when we look at ATP, we've got an ion repulsion, which does what to the energy level of ATP? So what we're doing is if we've got an energy diagram and we're saying here ATP to ADP, we just said it was even. Okay. What about ATP makes its energy go up? What about the products with ADP makes its energy go down? So that we end up with a strongly exothermic reaction. So what destabilizes ATP? We've got this ion-ion repulsion. Oh, light is not a good thing. Okay, we got an ion-ion repulsion which increases the energy of ATP. So that is now going to go up, making this reaction exothermic. What else contributes? So why are our products more stable than ATP? What about ADP and the phosphate actually drop the energy of our products? This was in our sapling homework. If we look at the free phosphate, that free phosphate has a lot of resonance stabilization. We lose some of that in ATP. Okay, so a lack of resonance further increases the energy of ATP. Increased resonance stabilizes the products and drops that lower. Okay. What else could potentially happen on that? This goes back to all your thermodynamic stuff. So our enthalpy isn't going to help us out too much because you don't need to know all the bond energies. So it's not a bad idea, but I don't think any of you know the bond energies. Or if you do, I don't. So what else?
Negative delta G.
So a negative delta G is going to be important. So that is an important aspect. We are releasing energy, so we do end up with a negative delta G, which makes it a spontaneous reaction. Okay, but the question we're asking is, why is it a negative delta G? Why is it that when we look at the energy diagram for this, what we're seeing is ATP is so high in energy, wow, or APD, ATP is so high in energy, and why ADP and the phosphate are so low in energy. So why is that the case? So one reason was we had that ion repulsion in ATP, okay, which increases its energy. One option was looking at the phosphate group, saying the phosphate group had resonance stabilization, which lowers the energy of our products. What solvent are we in? Water. What can water do?
Hydrogen ions.
It can hydrogen bond, okay, with oxygens. ATP does have a lot of oxygens, but they're all kind of buried within the structure. If we look at the phosphate, the free phosphate chilling on its own, I can put water all the way around that guy. What is that going to do to the charge of the phosphate? It's going to lower it, which stabilizes it, which then further drops the energy of our products. Okay, so we can look at hydrogen bonding. And then everybody's favorite, since nobody suggested it, all right, let's see if we can draw a hint here. We have Gibbs free energy. You mentioned enthalpy.
You mean, are you talking about entropy?
Entropy can also contribute to this. Okay. How is the entropy going to contribute? So we're trying to make this delta G more negative. What should the sign on our delta S be, our entropy?
Positive.
There should be a positive value. So entropy needs to increase. Take a look at the reaction. ATP to ADP plus phosphate. What happens to your entropy? It increases. Okay. So we do see an increase in entropy, which is also going to then further separate or make our delta G even larger, okay, or more negative. So all of those things are contributing factors to ATP actually releasing energy in this process. Okay. Questions on that? So, glycolysis. This is a nice little summary. I think I like it just because it's a nice little summary. We broke it down into glucose to fructose 1,6-bisphosphate as part 1, if you will, of glycolysis. And then we did fructose 1,6-bisphosphate to pyruvate as part 2. So we broke it into those 2 individual sections. The important aspect to note within this is what is happening as far as energy consumption or destruction. So if we go through and look at just glycolysis beginning to end, we produce 2 ATP. Okay, we had to consume 2 ATP, but we also produced 2 ATP. The only way that that can happen is that we have to get another phosphate into the reaction somehow, okay, which is shown on this slide as that 2P. That phosphate is a free phosphate floating in solution. We're looking at what is typically referred to as an inorganic phosphate. So somehow or another, that phosphate has to get connected to our structure, which is one of our important enzymatic steps. We'll talk about that in a second. Yes?
I was going over the lectures yesterday, and I got a little confused when you were doing glycolysis. Glycolysis produces 2 pyruvate.
2 pyruvates. Yes.
And each pyruvate produces 2 ATP.
The formation of 2 pyruvates, yes, that's right. It's not pyruvate per se, it's the fructose 1,6-bisphosphate. When we cleave that, We turn that into 2 molecules. Okay, that's the glyceraldehyde, and I think it's DHAP. The DHAP turns into glyceraldehyde. We have now 2 phosphates on those structures. Okay, those phosphates can go through and end up, in the conversion into phosphate, can produce a total of 4 ATP.
Okay, so it's not that 1 2 equals 2 ATP, it's that the whole process?
The whole process,
Okay.
yeah.
So you just said that glycolysis produces 2 ATP?
The net is 2 ATP, yeah. The net is 2 ATP because we used
Okay.
2 ATP to start with.
Why does it say 4 ADP plus 2 phosphate?
Free phosphate.
So you need the 2 free phosphates in the reaction. That is a very important step in the overall mechanism. If you don't pick up a free phosphate, if you pick it up from ATP, you end up getting no net production of energy. Okay, so there's something very special about picking up a free phosphate, because that free phosphate is ultimately what's giving you your 2 ATP production. Okay, and we'll look at that. The other big thing that we get out of this is we do generate NADH. NADH can later be shuttled into, where did it go? Into our oxidative phosphorylation to generate more ATP.
So does that go specifically to complex 1?
That would specifically react with complex 1, yes. We'll talk about that in a little bit too. So within this, glucose to glyceraldehyde, what are the major changes? So this is pretty much the end of our first step of this.
You need energy to produce
The
fructose
bond
corn syrup.
So we're really looking for our bond breakage. So what bonds are breaking? I should not have stood up. There's a help for referencing. That's 2, 3, 4, 5. What bonds break? Our bond between 3 and 4 is a big bond that breaks. What else? If we just break that bond, we still have a straight chain with all of those carbons and oxygens connected to each other.
Between the oxygen and carbon 1.
Also known as the anomeric position. Okay. So those are our 2 major bonds that go through and cleave, which is ultimately how we can then get into our glyceraldehyde 3-phosphate. It's not going to cleave beautifully. We have to go through a sequence of other steps to help facilitate that reaction, and that's what that first sequence of glycolysis is about.
Are you expecting us to understand that mechanism, or just
Um,
what the bond cleavage
Do
is?
not believe so. We're focused mainly on the bond cleavages. Are there other things that absolutely happen within that?
Oh,
Very
wow.
sure. There's a lot of things that happen within that to get us to the point where we can cleave those 2 bonds. Okay? But those 2 bonds are our key ones to go through and get us to that structure. Our first step is to phosphorylate. This is going to end up increasing our energy. We're using a kinase in this case. What does the kinase do? It's not quite phosphorylation. If we just said phosphorylation, then we could call this a phosphorylase. It's not a phosphorylase, it's a kinase. There's a big difference between those. There's a hint. The kinase is important because of ATP. Your kinase is facilitating the transfer of phosphate via ATP. Okay. That's an important one. And it does put it on the 6th carbon. Okay, so it's again recognizing those differences and looking at those enzymes and seeing what they end up doing. Because the kinase one does show up fairly frequently. So we do end up putting phosphates on structures fairly often. Okay. Sorry, I got a cough again. What's happening in this case? Looks like a bunch of crazy stuff is going on here. All we're really doing is isomerizing. We're taking this from, whoa, yeah, I knew I was going to do that. We're taking this from ultimately a functional group that is an aldehyde, and we are converting it into a ketone. Okay. That chemistry is kind of fun to look at, so if you're interested in that, you can talk to me after class and look at that, but not, again, super relevant. It is really relying on the fact that we are doing, or the fact that we have this hydrogen. That hydrogen relative to the carbonyl is known as, there it is. Go ahead and say it. This is our enolizable hydrogen. That hydrogen is relatively acidic, okay, which allows me to form the enol form of this structure. That enol form is then what allows me to do the keto, or not, sorry, our keto-enol tautomerization. That's what allows me to go through and do that conversion. Okay? So it is really, really relying on that enolizable hydrogen. So your enzyme has to come in and pluck that hydrogen off, and then whether that's exactly the same hydrogen that comes back on or not, who knows? Okay? But that hydrogen needs to be removed, and that's what's facilitating the isomerization. Okay. As an interesting note, you'll notice that carbon number 2 was oxidized. Did we use an electron carrier? No. Why not? What happened to carbon number 1? Carbon number 1 was reduced. Okay. The net is we've actually oxidized and reduced within the structure. We do not need an electron carrier because all we're transferring is hydrogens. And not hydrides, hydrogens. Hydrogen ions back and forth. So within this enzyme, we do not need NADH or FAD. We can take a look at this reaction. This is also a kinase reaction. How do we know it's a kinase? Fair enough. Let's try that again. What type of enzyme is this one? We know it's a kinase. We know it's a kinase because we are using ATP to put a phosphate group on our structure. Okay. That's what's really contributing to this. So we do have another kinase. That helps set us up for the next sequence of our reaction, which is ultimately cleaving these two structures away from each other, which is again going to rely on enolizable hydrogens. If I can find them right. Because we've got ultimately a carbonyl here. Yep. Yeah. I believe it's that hydrogen. Okay, so you could try and go through and play around with that enolizable system. Since we're looking at an enolizable transfer, we're not doing oxidation and reduction again. Okay, it's just the cleavage of that structure open. So we do not need NADH for this process. It does require your amino acids to facilitate and help kind of break and shift things around.
So does the dihydroxyacetone, that one enolizes, right?
The dihydroxy phosphate, yes, there it goes through another isomerization. Okay. Notice again we're using an isomerase because that's all that's happening. We're just changing the location of the carbonyl, which is again going to rely on your enolizable hydrogens, okay, facilitating that transfer.
From your lecture on this, you said that step 2 was an oxidation redox. I understood you today to say it's not an oxidation redox because of the enolizable hydrogen.
So, is the one?
No, it's step 4. It's when you were on.
You just said step 2.
Yeah, step 2 of this one, the dihydroxyacetone phosphate. Going from that, in your notes, you have the carbonyl.
I see your notes.
You have the carbonyl needing to become an oxygen and
So it's the aldolase.
the methyl becoming a carbon. So you said
Yes, so we are doing to convert what you're looking at there. Let's make sure I'm seeing that right. Yes. What you're talking about there is this step, trying to convert the dihydroxyacetone into glyceraldehyde.
Right.
So if we look at this carbon, what happened to it in the course of the reaction?
It It was reduced.
It was reduced. This carbon,
It was oxidized.
oxidized. Because one position was reduced and one position was oxidized, the net is the
Zero.
0.
Okay.
We didn't need an electron transfer agent, which is why when we look at this enzyme, you'll notice that you don't see NAD plus or FAD. Okay.
Because it's
So
within the.
it's within the structure. So by changing the atoms around,
Changing
you're just changing the location of what atom was oxidized versus
Which is
what
an
atom was
isomerization.
reduced, and that's ultimately just an isomerization. Okay. So if we look at the individual carbon, yeah, the individual carbon was reduced, but another carbon within that exact same structure was oxidized. Because we can couple those two with each other within the structure, we're not looking at a redox-mediated reaction. So when we talk about redox, really what we're referring to is the mediation. Do we need NADH? Do we need FADH?
That's right. It was one of my questions I asked anyway. Thanks.
So now that we're in the glyceraldehyde 3-phosphate, we now have our smaller molecule, and you'll notice that we have 2 of these now. Okay. One came from the dihydroxyacetone, the other one was directly formed as glyceraldehyde when we did that cleavage, the aldolase. Okay. Which means 2 phosphates, I can potentially make 2 ATPs. But if that's all I did, the net result is nothing, because both of those phosphates came from ATP. Right? What? Where am I? Oh, because I summarized. Yeah. Oh, man. I swore I fixed that slide. So let's just jump to here so we don't see that nasty little slide. We end up using glyceraldehyde 3-phosphate. We react a free phosphate from the solution, and we can put that onto the structure. So what we need to do is evaluate what things changed within this. All right, where did all the chemistry occur in this reaction? About carbon number 1. Okay, what was the result? Carbon number 1 was oxidized. There's nothing else going on within that structure, so we need a redox mediator here. That's what our NAD plus is doing. Okay? So
And
we'll,
the free phosphate.
and we'll need the free phosphate group. Okay, but we need them at different stages. The first thing we need to do is oxidize down our aldehyde, because we need something to connect to the phosphate. Because our phosphates are all oxygens, okay, we need some, or because our leaving group on our phosphate is only going to be water, we need something to attack that phosphorus. That needs to be an oxygen from carbon number 1. So the first thing we go through and do is do the oxidation reaction. Once the oxidation reaction is done, we can then facilitate the picking up of that phosphate. This all goes through coenzyme SH, okay, or cysteine, okay, which is very similar to coenzyme A because they both have that free thiol. Okay. What we're looking at here is a dehydrogenase. Why is it a dehydrogenase? You're removing H minus, and the other big aspect on this is we're dealing with that phosphate as well. Okay, so those are 2 big tips for our dehydrogenase. Yes?
Is this that organic phosphate that I pointed out earlier when I said 4-ADP plus 2-PI? Is this
Yes, this is the 2-PI. Okay. Why do we need 2-PI?
because you have 2 glyceraldehyde?
There are 2 glyceraldehyde 3-phosphates. So this reaction is run twice because there's 2 of these pieces. Okay. Once we've now got that structure, we can now transfer phosphate over to ADP. Okay. Again, we're going to end up using a kinase. Why? We're looking at the transfer of phosphate with ATP or ADP. So that kinase pops back in with this overall reaction.
So, on the test,
Mm-hmm.
you're going through a lot of systems. So are you going to show us the
Reaction?
structure, and then ask us multiple choice based off of the structure?
Yep.
Okay.
So one thing that we keep referencing is looking at the enzyme names. Okay?
Mm-hmm.
What does a kinase do? So we have to evaluate what's happening across our reaction, and then we're tying that to the kinase name. What happens across the reaction, we're tying that to a dehydrogenase name. What happens across the reaction, we're tying that to, I need another one. An isomerase. Okay. It's a good one, I can't remember them all. Okay, so what we're trying to do is see what's happening within those structural changes. So there's two approaches to how you can answer questions. One, I'm giving you the reaction. Evaluate what changes. I know an enzyme that does that change is some type of isomerase. Two, I give you the reactions and the names of these. If you've gone through and memorized the cycle, guess what? You know the name of the enzyme, there it is. Okay, so I'm trying to set it up in such a way that if you go through and strictly memorize, which is typically how it's taught, you can get the answer. If you go through and evaluate what things have changed and tie that, you can still get the answer. So I'm trying to provide two methods for that process.
One of the quirky ones is the dehydrogenase.
The dehydrogenase is
Because it doesn't really lose a hydrogen, it means add. Because they found adding it first, you were talking about this when you first went through the slides.
So it's not adding a hydrogen. It's removing a hydrogen. You're removing hydride from that first structure. Oh, Ben, stop.
So can you clarify the dehydrogenase, because on one of the, I can go back through it after class, but on one of the lectures you talked about the name really being the opposite of what it is because they located it first and so they called it.
So that is something that does show up in certain cases where they're referencing what they saw in those individual processes. So it depends on the overall reaction that you're looking at on when they went through and named it. So we'd have to go back and find that specific reaction. It's somewhere in the citric acid cycle.
But you're not going to after
No.
Okay.
Our mutase. So I want to kind of pick this up a little bit. I should have deleted that slide. Our enolase should have deleted that. Well, this is important because again we have that enolizable hydrogen. That's kind of what's facilitating our reaction. And we end up at the very end of this with pyruvate. Here's our last formation of our ATP. This is where we pick up our net. Okay, our net gain of 2 ATP. And again, you'll notice the enzyme that we're using is a kinase, because you're transferring a phosphate through the ATP mediator, if you will. Anaerobic metabolism. Why do we go through and deal with the anaerobic metabolism? If there is no oxygen present, can we do the electron transport chain? No, which means we don't get ATP. That's a bit problematic. So if we've got a system that only drives to oxygen reduction, we're going to run out of energy pretty quick. So we need some other way to produce ATP. Okay. Well, citric acid cycle produces one equivalent of ATP. Almost all of it is electron carrier mediators. So citric acid cycle doesn't help us out either. So we need to do something where we get ATP. Well, where did we get ATP? In glycolysis. That was the only place where we did that. Okay? But to do that, we had to convert NAD plus into NADH. Okay? So at this point, if we have no more oxidative phosphorylation, we have a surplus of NADH. We have no NAD plus. Everything shuts down. Okay? We don't get any ATP production. So what the cell has gone through and done, or what biology has gone through and done, is come up with an alternative way to produce some ATP as almost like a trickle current to try and just maintain. To do that, you need to have NAD plus so that you can maintain the cycle. Okay, so you can maintain, not the cycle, glycolysis. Okay, so we can just start cleaving the crap out of glucose molecules and hope we can generate enough energy to maintain life until we get oxygen back and we can reform or restart oxidative phosphorylation. Okay? That's what our anaerobic metabolism is all about. We use it as humans into lactic acid. Okay? The whole point of this is to form NAD+. So we sacrifice an NADH molecule. I was given numbers earlier. NADH produces what, 2.5 ATP? Okay. 2.5 ATP. So we are sacrificing 2.5 ATP from the electron transport chain to reform NAD+. What's the trade-out? The whole point is that we can then do, let's see, is it on the next slide? Oh, what? Apparently I deleted it. Oh my gosh. Fine. I'll just go all the way back. No, too far. The whole point is that once we reform that NAD+, that NAD+ can be used up in that previous step in glycolysis to reform ATP. Okay, so we sacrifice 2.5 future ATPs in forming 2 ATPs, okay, for that single NADH. Okay. That does convert some of our pyruvate costs, and it's not just a 2.5 for 2 conversion, because pyruvate ultimately goes through into the citric acid cycle and produces a crap ton more NADH. Okay. So we are actually sacrificing a larger amount of ATP possible production, but we can't go through the citric acid cycle if we don't have the oxygen. Okay? So that's the whole point of anaerobic metabolism, is to come up with some alternative way to get ATP. I'm going to find back where we were. This is another dehydrogenase enzyme. What you're looking at is that NADH being involved again in this overall process.
And this is where it's adding the
And this is where it's adding hydrogen. Oh yeah, here it is. How did they likely discover it? They probably discovered it doing the reverse.
Right, the reverse reaction, so removing the hydrogen.
So that's why they
here,
called it a dehydrogenase, even
even
though it's really a hydrogenating. Right? So,
well, because it is dehydrogenating lactate in the reverse.
in the reverse it is. In the reverse, it's really
So
doing
the
a
name-
dehydrogenase. So, and that's one of the
Describing
issues
reverse
is
reaction.
when are we using it? We're not using it to do the reverse.
It's not pyruvate.
We're doing it to get rid of the pyruvate.
Right.
Thiamine is cool. That's an important one to look at. Why do we say thiamine was cool? In this very first step, what is it doing? It is acting as a nucleophile, so an electron donor. And in the next step, what is it acting as? An electrophile. Okay, it's acting as an electron acceptor. Okay, kind of a cool molecule in that respect because it can both donate and accept electrons. We're looking at what was known as a carbene in organic chemistry.
Is there a mechanism for that?
No,
Do you want us to know just that thiamine acts both as a nucleophile and electrophile?
that would be a much easier question for me to ask on a test. Yes. Electrophile.
Give us a flat mechanism.
Yeah, no. I forgot what I was going to say on this. So all we've really gotten through is up to our glycolysis with our pyruvate. Pyruvate's converted into acetyl-CoA. Acetyl-CoA dumps into the citric acid cycle. Well, actually, as far as slides go, it's not that bad. And then we've got, so this slide is actually kind of nice, though. I think there's a white box covering this. So if you download these slides, you may not see that image. Okay, this is kind of nice because it shows where everything is in the mitochondria and shows what's going on. Glycolysis is not happening in the mitochondria, it's happening in the cytosol. All those pieces then get transferred into the mitochondria. Okay, and the mitochondria goes through and processes it through your citric acid cycle. So we got the citric acid cycle showing the individual pieces, okay, or sort of our cycle happening within there. You'll notice that they are trying to show that process through complex 2 of your electron transport chain. The electron transport chain is really just there to generate hydrogens, or H+ on the inner membrane space, which can then be taken advantage of by ATP synthase to form ATP. We've got the big drawing all the way around of all the individual pieces, and you could go through and look at each of the enzyme names and try and see what they're tying to. Right? And I'm kind of out of time, so I'm just going to real quickly. Our pyruvate dehydrogenase complex, kind of a fun one. This one we're actually kicking off CO2 as well as removing that hydrogen. Okay. To get our acetyl-CoA. Whoa. The only reason I included this slide is because it does a nice job of showing what enzymes are involved in each of those enzymatic steps. Oh, that's really bright. and it also kind of helps you see where you're forming your NADH, okay, your electron carriers. You'll notice that in the steps that you've formed NADH, in fact, any of your electron carriers, what's the name of your enzyme? Dehydrogenase. So that dehydrogenase is really the one that's generating your electron carriers. Okay, so you can go back and take a look at glycolysis, and where we formed our NADH was a dehydrogenase enzyme. Okay. Our overall cycle, looking at each of the individual steps that exchange in between those. Okay, so you should be looking at those structures and saying, okay, what things change? Okay, what kind of enzyme would facilitate that change? So this is looking for those structural aspects and tying them to enzyme names. The other thing you can look at is how is it happening. For instance, acetyl-CoA acts as what with oxaloacetate to form citrate? This is a tricky one. If it's cofactor, it's not going to change our structure. So I'm not referring to coenzyme A, I'm referring to acetyl coenzyme A. How does coenzyme A change the structure of the acetyl? Turns the acetyl into a nucleophile, which is what's putting on your acetyl group. So again, looking for those differences in charges, how do those balance and how do those shift through, how do those change the structure? We are definitely out of time. Control. NADH is going to be our primary inhibitor for these. Why would NADH act as an inhibitor? The whole point of citric acid cycle is to make NADH, which you can then go through to make energy. Well, if you've already got enough energy, should you be doing NADH or making NADH anymore? So the NADH will come in and it inhibits almost all of those steps because we don't want more NADH if it's there in excess. Okay? One of the big ones that I'll note here is our citrate synthase. It is a feedback inhibition because it is a product. Citrate synthase is one that's actually kind of interesting that it's inhibited by. It's inhibited by our NADH, and that kind of gets bizarre because we aren't forming NADH in that reaction. So why is that one an inhibitor? If you go through, let's see, I don't think it's on. Oh, cool, it is. If we go through and take a look at our anabolic reactions, citrate has relatively little things that it's involved with, whereas oxaloacetate is involved in a lot of other processes. So why might I want to inhibit that enzymatic step? Well, citrate isn't involved in a whole lot of other things. Do I want citrate? No. If oxaloacetate is incredibly important in lots of other anabolic processes, I want a pretty steady constant concentration of oxaloacetate. So if that solution or that compound is missing, do I want to be converting whatever I've got over to the citrate? No, I actually want to stop that process because the citrate's not going to do anything helpful for me. The oxaloacetate's going to be more important. Okay, so that's why that enzymatic process is controlled by your NADH, because we don't want that process continuing forward because oxaloacetate is really important. Electron transport chain, proton gradient, yada, yada, yada. We've got all those enzyme complexes all tied together through those different complex systems. Ultimately, our big idea is to transfer electrons from NADH in complex 1 to coenzyme Q. Complex 2 transfers electrons from FADH2 into coenzyme Q. Coenzyme Q takes those electrons, shuttles them to complex 3. Complex 3 shuttles those electrons to cytochrome C. Cytochrome C then shuttles those electrons to complex 4. Complex 4 is our actual big end step of our reduction. We convert oxygen gas into water. Each of those steps does a transfer of hydrogen ions. Wow, that last few minutes went by really fast. ATP is kind of neat. Two big components, F1, F0. There's our ATP yield, 30, 32. Sorry, done.